Hey everyone, welcome back to the EduHaven Done with Pencil series. This time we're going to go over AP Biogenetics. So without further ado, if you're ready to succeed, let's go. So the first thing we have to talk about with genetics, of course, is DNA. DNA. So DNA is called deoxyribonucleic acid, right? So deoxy, there's no, not okay. So the monomer, if you haven't watched the biological macromolecules video, the monomer for DNA is a nucleotide. So what they look like, they have three parts. They have a house, it's a 5-carbon sugar, because it looks like it's 5 carbon. There's shown there's the carbons are here, by the way, but you don't draw them in here. Then after the 5-carbon sugar, you have, or the house, you have a pool or a phosphate group, right? And then you also have in the maybe the front yard, you have a basketball court, a base, a nitrogenous base. And this is the most important part, because the thing about the nitrogen base is that they can change. And that's what gives DNA the energy storing, like it allows it to store information, because there's 4 different types. There's, I'm not gonna write them out, but it's adenine, guanine, thymine, and cytosine. Now, with the first two and the last two, the first two, remember Andre Agassi. So he's a very pure tennis player. These are called purines. They have the 2-ring structure, and these are called pyrimidines, thymine and cytosine. They have a 1-ring structure. And also another thing about these is that they pair up. So what it allows it to do is it pretty much makes it so it always has it's a uniform like width. So that's why that's really important. Remember, adenine goes with thymine, guanine goes with cytosine. All right. 5. There's the 5' carbon. So DNA always goes from 5' to 3'. That's how it's built. So what this means is these strands are actually, so when you have DNA strands, it's a double helix, and it's anti-parallel. So one is going to go 5 to 3, while this one goes 5 to 3. So it's anti-parallel. And also, when you do replication, both are used as like templates. So that means when you have new like DNA, it's semi-conservative. So half of the DNA remains the same. So let's say if I there's a thing called radioactive isotopes. If I labeled the radioactive isotope in here, like this side right here, or yeah, you can't really label sides of both of them, but let's label these with the radioactive isotope blue. That's what it's called. And then when you do when you do replication, what ends up happening is you end up now. It's now half of these are always like they're always there. It still has the labeled the blue. Blue is still labeled. Now you do it again, and it's gonna be this and this, and then you can keep going, but you can't get rid of the blue unless you, I guess, I don't know, maybe, yeah, you can't really get rid of the blue. And so, let me go back to this one last thing. So, with DNA, so it's semi-conservative. With DNA, you have to remember that, so these are the phosphate sugar on the side. It's phosphate sugar, phosphate sugar. So it's like an alternating pattern. In here, these are the bases, and these the order in which these go determine. So the central dogma, of course, is DNA to mRNA to polypeptide, pep, and then it goes to the trait/phenotype. So that means the DNA, the order of the nucleotides or the nitrogenous bases, determines the different polypeptides you use in different traits. But because there's 4 of them, it means you can have like many different like combinations. And there's only 20 amino acids, so yeah. Now, so it's always in it's called a frame. It's 3. Or a codon. It's 3, so that means 4 times 4 times 4, 64 combinations. So you also have a little bit of redundancy in the codons and the amino acids. But one last thing we have to go with: bonds. So along this backbone, there are covalent bonds. Now, these covalent bonds are a specific type of covalent bond. It's called a phosphodiester bond. And within these, there's also a bond. These are hydrogen bonds. So these are much weaker. If you remember the biological macromolecules video or the properties of water video, these are much weaker. So what that means is you can actually, it's a very stable molecule, but you can not cut, but you can remove these to form a sort of bubble. Now, what are these bubbles used for? Replication and transcription, critical processes for DNA, which we'll go into later. The next topic is DNA replication. So I want to go back to the central dogma of biology. Now, what you need to know about this stuff. So there's also another step in here. DNA has to get replicated, and that's what we're going to talk about right now. That was kind of a big circle, or a loop-de-loop. I'll just do that. So the interesting, so DNA, there are 6 enzymes that work with it. Helicase, which unzips, so it unzips, it breaks the hydrogen bonds between, which are weak, which why DNA is a good information molecule. It's a weak molecule, so therefore it's able to break the hydrogen bonds and therefore make bubbles, replication bubbles, or replication forks, and now it's able to be replicated. Then we have, there's 2 of them. It's topoisomerase and DNA binding proteins. These kind of stabilize the DNA molecule, prevent knots, prevent converging. But those are 2. They're not like that important. We don't really need to know them. Then you have RNA primase. You have DNA polymerase. And you have ligase. And we'll get into these 3 last ones because they're the most important ones, I would say. So we know that I probably shouldn't erase now, but so I'm gonna draw a replication fork. So let's say this is one bubble, this is another bubble, and we're gonna label them. So here, if they give you this information, let's say we know that this side is a 5 prime. We know that this side is a 3 prime. That was a little messy. A 3 prime. Well, if we know this one side, we know the other side, right? Because it goes 5 prime, 3 prime. That means it's also anti-parallel, so that means we can also figure out that this side is a 5 prime and this side is a 3 prime. Now, what this means is that when you when you replicate it, the new DNA, and it's going to be on the inside, it's going to be 3 over here, 5 down here, 5 over here, and 3 down here. And so that's going to be the new DNA being created. And so the way it works is it goes down in two directions. So what my teacher suggested when I learned this is you put a little dashed line. This is like the center, and we know that. So this would have to be 5, 3, 3, 5, opposing these two, of course. So what happens is the first one of the bottom three was RNA primase. So what RNA primase does, so what you have to understand is that the next one, DNA polymerase, it creates the new DNA. However, it can't just start building stuff. It needs RNA primers or like small bits of RNA to build off of. And so that's what RNA primase does. It will create small strands. However, there's a little bit of nuance to this. So the way it works is there's two types of strands: a leading strand and a lagging strand. So the leading strand is the one in which the direction is 5 prime to 3 prime outwards. So this out, so out, oh my god, outwards. So let's see. This is 5, 3. Okay, so that means it's going to be building from 5 to 3. Remember the inside one we look at. Let's look at the bottom one. 3, 5. So that means, whoops, that's fine. That means it's going to be building out this way. So this one is called a leading strand because the leading one, it builds outwards, out from like the bubble. So this is called a lead. Let's label it lead. Lead. Now, this means this has to be a lag because 5, 3, it's building towards the center of the bubble. So this is a lag. Now, from this side, 5, 3, this is going towards the center, so it's a lagging strand, sorry. And this one is 3, and then it's supposed to be a 5, remember? Because it's, so 5, 3. That means this one is going to be building out this way. So this is a lead. And so what RNA primase does on the leading strands is it builds in where it starts. So it's going to build some RNA primers here. That's where the leading strand is. And this is another leading strand. So here, right? Now, where does the lagging strand start? Where does the start like replicating? And the answer is it's kind of in multiple spots. So it goes out a couple, then builds one. Goes out a couple, builds one. And it keeps doing that. Remember, these are much longer. There's, DNA is like thousands of nucleotides long. So, and the same thing happens over here. So it goes, builds out, builds RNA primer, builds out RNA primer. And so remember, the primers go on, they're kind of like fragmented. These are called Okazaki fragments, by the way, when they're built, like, when they're replicated this way. They're called Okazaki fragments. Okay. Now, you have RNA polymerase. So what RNA polymerase does is it builds the new DNA polymerase. I apologize. It's called it's DNA polymerase. It builds out. So it goes from starting from here. It starts building out. Da, 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 da. Now, an important thing to realize, so, is that these are like the opposite of the template strands. What that means is it will, so it's the opposite in the sense of, remember the ACG GT? It's the opposite of that. So let's say this is, let's say ATG. That means when you build it, it's going to be TAC. Now, the RNA primers, remember, RNA doesn't have thymine. It has uracil instead of thymine. So it's going to have, if this was over here, it would be UAC instead of TAC. It's UAC. But you don't, but yeah, that's another like important fact. So what this means is, now the same thing happens over here. So it builds 5, 3. So here, 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 here. Now, then it gets rid of the RNA primers, and a different type of DNA polymerase comes in and fixes the Okazaki or it fixes the Okazaki fragments, of course. So it puts these in, but it also replaces the primers with DNA. Now, sorry, it's not going to work with my little demonstration. Now, you might realize that these primers, they're not really bonded together. So what happens is the ligase ligates, which means connects. So the ligase comes in and will connect and create the little bonds between these, the phosphodiester bonds. So now we have a replicated DNA strand, and these replication forks happen several times, and they all go out, or they all push out in this way, and then they'll all converge. They'll all they'll all meet, and then you'll have a replicated DNA strand. And now this one has this one's a new DNA strand. This is a new DNA strand. So that's how DNA replication works. The next step, so we have the first step, replication. However, DNA to mRNA is another step. Now, this is called transcription. So transcription is DNA to mRNA, and it uses a different set of enzymes. It uses, the main one is RNA polymerase instead, which is why I kept saying RNA polymerase earlier. So, sorry. So we have, I'm gonna make a big one. So with transcription, unlike DNA replication, it only goes for after certain genes. So let's say this is the gene of question. Let's call it gene X. So in order to get gene X, you need a DNA polymerase. Now, DNA polymerase, how would it know to or RNA polymerase, how would it know to specifically transcribe this stuff? So it needs a certain spot to attach. It has a promoter. Now these promoters are actually highly conserved, because of they have a thing called a Tata box. Now these promoters can also be regulated, but we'll get into that later. So we have DNA polymerase. That's called, I'm sorry, I keep messing up my words. I mean RNA polymerase. That's why it's called done with pencil, because it's highly scuffed. But information is still good. So RNA polymerase. So RNA polymerase comes in, attaches to the promoter, and will keep going. And it will keep going until it hits it's a multiple A sequence. So there's multiple A's, but you'll keep going. So it goes over, creates mRNA. This is the mRNA. Remember the two strands? This is called a template strand, and this is this. Remember this strand. So the opposite strand, it's kind of similar, except remember it's mRNA. So that means it's gonna be uracil here instead of thymine. So it's the same exact code, except it's uracil instead of thymine. Now we have this transcription. We have this what's it called? We have the mRNA. So after mRNA, after we've done all this, mRNA, you kind of have to with eukaryotic eukaryotic cells, it will in the nucleus, because it has a nucleus, it will add to the 3' side, it adds a poly A tail, and to the 5' side, it adds a chemical cap. I think it's a G cap, they call it. They had a G cap, and they have a they had a poly A tail. So, but then within this, there's these things called introns and exons. It splices or removes the introns. So let's say that's intron, that's intron, that's intron. So then they reconnect it, and now we have the final piece of mRNA, and it gets sent over to a ribosome. So now we have a ribosome. Now the ribosome has two parts, and this process is now called translation. So a ribosome has a large ribosomal subunit, so large ribosomal subunit, and it has a small ribosomal subunit. I abbreviated them. So the mRNA will come over. We're going to use blue for the mRNA. So the mRNA will come over, and so these units, the large one, so the ribosome will have an E site, a P site, and an A site. And for the first one, the subunit, it does initiation, the process of initiation, which is when the mRNA comes in and it starts at the P site. And what happens at the P site, they have these things, there's codons. So every three nucleotides, every three bases is a codon. So that's a codon, that's a codon, that's a codon. And what happens is these codons are like the bases. So now it will come in the P site, it will initiate. This will always, you know what this is. This is, you always know it's starting. It's A, U, G. And the reason why is because it's called the start codon, or methionine. I'm not going to spell it. You just need to know it's the met amino acid. Now it elongates. So the ribosome will shift over. So it will shift over. Let's pretend it shifted over because we don't have graphics or animation. So it will shift over to the E side. And it's going to eject. It's going to get ejected. Or for the first one, it won't eject because so now after it goes in the P side, it goes in the A. The next ones will go in the A side. So then it shifts, it translocates, what it's called, and then at the E side gets ejected. So it's not needed anymore. But there's an important step with elongation. There's another type of RNA. So we know mRNA. That's the messenger RNA. This is called rRNA, which is ribosomal RNA. That's what the ribosomes are made out of. Next, the last RNA that you need to know is called tRNA. It looks something like this, and then it has the little anticodon. The anticodon, like the name suggests, is like the opposite of the codon. So it needs to match up. So if we have the AUG over here, it needs to be the UAC, the UAC anticodon, and it will bring in the MET, or the amino acid that it corresponds to. So these come in, and they'll hit over here, and they'll bring the amino acid, like that. And then the ribosome translocates. The next amino acid, the next tRNA comes in. It makes the peptide bond between the next one. It translocates again, ejects it, and that process continues. This is a process that's called elongation. This process continues until it hits. There's 4 stop codons. You don't need to know them. I think it's UGA, or maybe UAG, UAA. It's something like this, but you don't need to know them. They're not important for this course. But then after that, you now have a protein over here. That's not a protein. This is a protein. So now, remember, this will keep going on, or it's really a polypeptide. If you want to get specific, it's polypeptide. You get hundreds of them. And now this polypeptide will do some activity, or it will run some process that expresses a trait. And that's how the central dogma really works. Alright, so that is part 1 of the EduHaven Done with Pencil AP Bio series on genetics. Thanks for watching. I hope you enjoyed it, and I hope you succeed.